I mean, we are really living in a time where a single sort of data layer or type of information is often not sufficient, right, to learn about human biology, because it's a complex interaction maybe between different molecular classes, but also how sort of underlying genetic variation influences the functionality of processes. And of course, GPCRs as one window to human biology, as a regulatory aspect of human biology, is an important sort of turning point or tuning point, maybe, that I guess we haven't really studied in our approach yet, because we decided to go with the canonical sequences. So we sort of provide a baseline information set. But yeah, it will be quite exciting to see how other techniques will sort of add value to what we have started. Look at sequence variation in terms of interaction, sequence variation in terms of ligand binding, and probably also, using orthogonal methods like mass spectrometry to look at structural elements of the GPCRs.
Mm-hmm. Yeah, I think that is really important. And it occurs to me that we often talk about binding reactions in chemistry. There are two elements, right? And you get a substrate enzyme complex, for example. You try to reduce everything to two elements. But of course, we're talking about complex interactomes here. And so you need multiplex methods to try to sort all of that out. And it brings to mind in the GPCR space that there are still receptors in the body where they're expressed, the genes are there, they're expressed in a tissue-specific way, they're on cell membranes, but we don't know what the molecule is that actually activates those receptors. Those are called orphaned receptors, because we still don't know what the natural ligand is for those. And one of the possibilities is that the receptor itself needs to have another protein to form a complex to actually be competent to bind a ligand, that is its natural ligand. So this is an opportunity to test that hypothesis for deorphanization programs that are very important for drug discovery. And then if you find a putative, say, receptor ramped complex that now becomes the active receptor for a new ligand, you can validate that with some of the other technologies, for example, MolBoolean, which allows you to definitively show that that complex exists and could be competent to be the actual receptor for a new drug entity, for example. So I think these omics methods are important. Protein-protein interactions are important. Defining them in time is important. And there are a lot of subtleties to it, as Jochen suggested, because of sequence variations, post-translational modifications, and so forth that could be interrogated in these methods.
Yeah, I mean, it's interesting to just think. I mean, we learn a lot about this, proteome form space, which expands, of course, the 20,000 canonical protein encoding genes that we have. But also, I mean, and I think that we sort of touched upon a little bit in our study, is that even if you can confirm on a, let's say, co-expression RNA sequencing level the coexistence of the receptor, of the two receptors, we actually show experimentally that doesn't mean that they do bind, right? Mm-hmm. So it's not only looking for the positive hits and sort of build a library on that, but also to reconsider that despite co-expression, maybe interaction is not present. And that, of course, we can then expand to different tissues and look at on a molecular level or on a cellular level using other methods like MolBoolean. Can we confirm this? Can we sort of, support or challenge our hypothesis? So I think that's quite an interesting sort of direction to think.
Mm-hmm.